Wine, and I'll be talking about HPV. So, what is HPV? HPV, or human papillomavirus, is a group of viruses known for causing warts, with some high-risk HPV being carcinogenic, meaning that HPV can develop the infected cells into cancer cells. HPV targets interferons, or IFNs, causing prevention of IFN response, creating an environment that promotes cancer. This is important as interferons are responsible for antiviral mechanisms and help to create a pro-inflammatory environment. HPV can spread through intimate skin-to-skin contact. When this viral infection starts, HPV infects keratinocytes located in the basal layer of the cervical epithelium. Keratinocytes are a part of the innate immune system and are non-professional APCs that can induce the expression of Th1 and Th2 type cytokines, as well as cytotoxic responses in both CD4 plus T cells and CD8 plus T cells. In the female genital tract, keratinocytes express TLRs that promote the production of these cytokines, which will cause inflammation in areas with infected cells. However, HPV is able to modify cytokine levels as a means to evade the body's immune response. Through downregulation of IFN pathways, HPV is able to avoid Langerhans cells, which is a subset of dendritic cells.
Now we're going to talk about how HPV and the innate immune system interact. So keratinocytes, the outermost layer of our epidermis, they are known as non-professional antigen-presenting cells. They are cells that have PRRs, pattern recognition receptors. A type of PRR is known as a TLR. TLRs can specialize in different pathogens, but for HPV has a double-stranded RNA genome. TLR3 is able to detect a double-stranded RNA genome, and this is known as a PAMP, the double-stranded RNA genome. The binding of this, therefore, stimulates the increase in inflammation, the recruit of dendritic natural killer T cells, which would stimulate the production of cytokines such as NFK-beta, IRF3, and TNF, which halts the abnormal proliferation of these cells. Dendritic cells aid with adaptive immunity, and natural killer cells are able to directly kill the virally infected cells. An increase in inflammation would therefore signal to activate TLR9. TLR9 would tell the keratinocytes to therefore secrete IFN, or interferon 1. This has several antiviral mechanisms which helps with the depletion of the virus. HPV can also escape the innate immune system via the decrease of expression of PRRs on keratinocytes, which therefore this interaction would happen a lot less. The decreased transcription of NFK-beta, TNF, and IRF, which also are cytokines which help stop the proliferation of these cells. And this would also decrease activation of the inflammasome, commonly paired with the activation of NLRs, a type of PRR which stimulates pyropoptosis, which is a fiery cell-mediated death. Along with apoptosis, the innate immune system works really hard to kill these cells.
It's me again. What signaling is happening in the innate immune system? Well, once the cells are infected, APCs such as dendritic cells will bind to PAMPs using PRRs and present these antigens to other leukocytes. These leukocytes are natural killer cells, otherwise known as NK cells and T cells. Since we aren't talking about the adaptive immune system, I will gloss over the small fact about the T cell's function in helping the innate immune system. Once a T cell is presented with an antigen, cytokine signaling will occur and recruit IL-15 to produce inflammation. IL-15 will also promote proliferation and promotion of NK cells. NK cells, once presented with the antigen, will be able to recognize infected cells and will kill them through either use of granules or through fast ligand causing apoptosis.
T cells and B cells are the two main components of the adaptive immune system, and provide cell-mediated immunity and humoral immunity respectively. So how does the adaptive immune system react with HPV? Well, viral antigens are presented to the immune system in a couple of different ways. One is when HPV gets swallowed up by a dendritic cell. The dendritic cell will present antigen through MHC class II to CD4 positive helper T cells. These T cells will provide cytokine signaling and support the activation of cytotoxic T cells. But the main way is through epithelial cells when they're infected. They present antigen through MHC class I to CD8 positive cytotoxic T cells. And when these cytotoxic T cells are activated, they identify and kill cells infected with HPV. However, HPV has adapted, and the way it avoids cytotoxic T cells is that it downregulates MHC class I with a protein called viral oncoprotein E5, and this counters the cell-mediated response and allows the HPV to continue to produce. So the vaccine has actually allowed B cells to be activated as well and provide a humoral response where neutralizing antibodies are created and bind to the virus and prevent the virus from infecting other epithelial cells.
HPV infection evades the immune system through various mechanisms, leading to infection and increasing the risk of cervical cancer. I will discuss the immune response to HPV infection, and then I will talk about HPV immune evasion mechanisms. On the left portion of the image, NK cells engage with HPV-infected epithelial cells. These cells act as first responders by recognizing PAMPs through PRRs, such as TLRs. MHC molecules are infected, then T cells are activated. CD8 T cells kill infected cells, and CD4 T helper cells help this B cell maturation and antibody production. Now we'll talk about the HPV immune evasion mechanisms work. On the right side of the image, HPV has viral oncoproteins such as E5, E6, E7 to downregulate MHC class I molecules, impairing antigen presentation and cytotoxic T cell mediated clearance. This helps cytokine networks, suppressing pro-inflammatory and antiviral signaling pathways like interferon responses. These immune evasion strategies contribute to the development and progression of cervical intraepithelial neoplasia and eventually invasive carcinoma. In this image, we show cervical cancer progression. The image is divided into 3 panels. The first one shows HPV infection as virus attaching to a human cell with arrows, indicating viral entry as asymptomatic, triggering an immune response involving T cells. The second panel displays the immune response with T cells, natural killer cells, and macrophages evading the area. And the third one shows cervical cancer progression, displaying reduced T cell activity, allowing for persistent infection. And eventually, a healthy cervix transitions into abnormal cells and a cancerous mass.
An infection of human papillomavirus has a range of outcomes from harmless infection to cancer depending if the infection is from a low-risk strain or a high-risk strain. The prognosis depends on whether the infection resolves on its own, causes persistent health issues, or progresses to cancer. The general prognosis is that HPV infections are usually asymptomatic, and the immune system usually clears up the infection within a couple of years. For low-risk strains like HPV-6 or HPV-11, these cause genital warts but is generally treatable. Low-risk strains don't usually lead to severe health problems. High-risk strains, on the other hand, are more likely to persist, and the longer they do, the higher risk of more serious complications. These high-risk HPV strains can lead to cellular damage to areas like the cervix, anus, or throat. HPV-16 and HPV-18 are the highest cause of cancer, especially cervical cancer when left untreated. With treatment, though, the HPV infection is usually positive, or the prognosis is usually positive. Precancerous lesions can be removed, and the cancer caused has a higher survival rate when detected early. Vaccination and close monitoring are the most effective strategies for positive outcomes and prevention of HPV infections and related cancers.
HPV has a variety of global and socioeconomic impacts that affect us till this day, as it is the most prevalent STI with more than 150 strains found in both men and women. This can cause cervical cancer and is mainly found in low-income classes. This might be due to decreased screening for HPV, decreased vaccination, decreased STI education, and decreased access to healthcare, leaving this population vulnerable. Risk factors include being a young woman and being sexually active. Ongoing questions about this virus is if you need a pap smear after vaccination. Is there a false sense of protection that the vaccine provides and women might be engaging in highly risky behavior? Are there vaccine programs that we can mandate for low-income classes to give them access to healthcare? And are the antibodies that are mounted against this virus from the vaccine correlated with the pH of the vaginal canal? Possible solutions for this virus is the mandated and regulation of pap smears in women, letting women know their HPV vaccination eligibility, and providing education on HPV in middle schools and high schools during STI training, and lastly, giving preventative methods such as condoms and other resources more readily to prevent the infection of future generations.